Welcome to episode 32 of How Positive Are You? Today we're very pleased to have with us David Rasnick. He's a PhD biochemist and longtime critic of the HIV AIDS dogma. He's a member of the board of Rethinking AIDS, organized the program for RA 2009, and was responsible for breathing new life into the organization Rethinking AIDS in 2005 and 2006. David graduated from Georgia Tech with a PhD based on studying protease inhibitors, something he continued once he graduated. I'm also here today with my co-host, Terry Michael.
Thanks, David. By the way, since we have two Davids here, David Crow will be David, and David Rasnick will be Dave. And Dave, we're really pleased to have you on this episode of How Positive Are You? But before we begin the questioning, I think that it's possible that David may have some feedback from our listeners. David?
Yes, I do. The first one is from Paul. I think he's in Lithuania or Estonia or somewhere in the Baltics. I'm a big fan of How Positive Are You? And I'm always looking forward to new episodes. The podcast is one of the main components of the HIV skeptics movement. However, I'm disappointed about the delivery. New episodes take ages to show up in my iTunes feed. I don't know if this is something you can change, but faster delivery through iTunes is something I hope to get for Christmas. Well, we did have a lot of problems with iTunes, and a few weeks ago we were able to solve those problems, and now the episodes should be showing up on iTunes just about as soon as we release them. So I'm very pleased to say that Paul should be happy now. Second feedback is from Emmanuel. I listened to the shows on How Positive Are You, and they're great. I think it's a good idea to give interviews to people like Dr. Scherdell, Dr. Juan Jose Flores, Dr. Heinrich Kramer, etc. Another place for feedback, those are received through emails, but our blog on howpositiveareyou.com with episodes 30, which was John Lauritsen, and 31, Maria Gracia Gonzalez, there was an extensive discussion amongst people who went to the site, registered for free, and then just started talking to each other. The discussion recently has been about poppers, which was stimulated by the John Lauritsen podcast. So that's all the feedback we have, and welcome to the show, Dave.
Thanks, David. It's a pleasure to be with you.
Yes, Dave, thank you very much for taking time to do this. Why don't we begin by asking Dave to give us a little background on how he got involved with the whole question of the mysterious retrovirus and the amorphous syndrome called AIDS. How did, professionally, obviously you have a professional background in the chemical side of this, but tell us how you became involved.
Yeah, it's a long story. I'll try to keep it as succinct as possible. I moved to California in September of 1980 in the Bay Area to set up my first biotech company called Enzyme Systems Products. And about that time, I started going to a Bay Area organization. It was called the Bay Area Peptide Group that met once a week. And then I started hearing about these odd diseases that were showing up only in gay men. I'd never heard of them before. I'd never heard of Kaposi's sarcoma or pneumocystis or cryptosporidium or any of that stuff. I'm a chemist. And this was before AIDS even had a name. But obviously, AIDS and what was going on among the gay men had been going on for some years. Back into the 70s, it started popping up. I believe Joseph Sonnabend has reported on that from his work in New York as a physician treating these gay men. So then I was very interested in that, especially after 1981 when AIDS became formalized in I believe it was Gottlieb down in L.A., reported 5 gay men who had no association with each other, but they came down with, I guess, Kaposi's and pneumocystis or something. I don't remember those details too much. But I do remember that it was very interesting to we scientists. I was a chemist, there was virologists, there was regular biochemists, there was biotech people, there were people at UCSF, and generally it was a melting pot of biologically oriented or interested professionals that met at this Bay Area Peptide group every week. And we'd have a speaker, but then after it was over with, we'd go out to a Chinese restaurant, and then we'd start hearing the scuttlebutt of everything that was going around. And that was the primary reason why we all got together, really, was for that socializing. But, this AIDS thing didn't go away. It just kept getting more interesting, and, what was it? It reminded me of the Andromeda strain, this book that was turned into a movie, I believe, back in the 70s or 80s. And I was thinking, this AIDS is my Andromeda strain. Very, very bizarre. Only certain people were being affected by it. And so we were all very intrigued. We all talked about it all the time. And then in 1984, as we know, April 23rd, 1984, it all sort of crystallized out with Robert Gallo's press conference where he unleashed upon the world what we now know as HIV. And at that point, I got really, really excited. I thought, okay, good. Now we know what's causing AIDS. It's a virus. It was HTLV3 in those days. And being so close to San Francisco and having, a few years of this Bay Area peptide group under my belt and lots of collaborations with people at UCSF and other places, I talked with some of the virologists about what kind of protease. Proteases are enzymes, and all enzymes are proteins. And they're probably one of the largest classes of enzymes. They process proteins. There's probably not a biological process that doesn't have a protease involved in it one way or another, from fertilization through development, immunology, food digestion, hormone processing, everything. Some protease or groups of proteases are involved in this, including pathology. Lots of proteases are involved in tissue-destroying diseases like arthritis, emphysema, cancer, and parasites, which I've worked on personally. It was very rewarding over the years doing that. It was non-political science, which was what made it so gratifying. And then, of course, viruses. I knew that viruses tend to have at least one protease in their genome that is absolutely essential to their replication. For example, the flu virus has a protease. I believe it's a serine protease that they require. So I asked these virologists at UCSF, what kind of protease would a retrovirus have? And they told me an aspartyl protease. There's 4 different classes, basically. Now there's even a 5th. But in those days, there were 4 basic classes of proteases. And it's like saying, is it a Volkswagen, a Jeep, or a Ford, or something like that? And then once you know what brand you're dealing with, then you know all the models and everything else. Well, with these retroviruses, the protease was an aspartyl protease. So I said, great. Then to that point, I had never actually worked on aspartyl proteases, but it's to a chemist, they're all pretty much equally accessible. All you need to know is learn about that particular class, which I did. And then within, I'd say a week or two, I started designing inhibitors for aspartyl proteases. I was in my early 30s in those days, and I wanted to cure AIDS, win the Nobel Prize, be carried on everybody's shoulders for solving a great, horrendous problem. And I stopped that, though, that whole exercise or activity, designing these inhibitors for HIV protease, from commercial reasons. My little company, Enzyme Systems Products, we only had, I can't remember exactly now, maybe 2 or 3 chemists in those days. And I had a friend, Jake Plattner, at Abbott Laboratories, where I used to work in the late 70s, and I knew he had a team of chemists, about 20 of them, that had been working for several years on a human aspartyl protease. So I realized he had probably a couple of thousand inhibitors already synthesized and in the bottle, so I dropped it. I realized there's no way I could catch up with.
Let me interrupt you here at this point and ask you, we're now talking about one of the 2 classes, I guess, of drugs that are part of the drug cocktails, protease inhibitors. Just before we continue with that discussion, the other class of drugs are the DNA chain terminators. My science background is not enough to explain it. Can you explain in the drug cocktails that came along in the 1990s the difference about in approach?
These viruses have certain machinery that they produce in their own genome. The retroviruses, they have three basic genes: the GAG, the POL, and the M. But the viruses typically require the host cell's machinery to reproduce themselves. And one of the enzymes that the HIV, these retroviruses have, they call these reverse transcriptases, and these enzymes that require nucleotides. And then there's inhibitors for those enzymes called nucleoside analogs, and that was AZT, for example, DDI3TC, D4T. The whole menagerie of nucleoside analogs went after that enzyme. Then in the 1990s, that's when the protease inhibitors came on the scene. That really became approved by the FDA, and they were designed to inhibit another of the enzymes, the HIV protease. And that's what I was working on for a very short period of time. The idea being that you stop the production of HIV by inhibiting its protease.
Do proteases always take apart proteins, or are they used to build up proteins, or do they do both?
You can go in both directions. Typically, they're cutting apart. They're like scissors, molecular scissors. They cut off little bits and pieces. Like think of the proteases in your digestive tract.
Right.
They chew up big chunks and make smaller ones out of it, and then there's other ones that even chew up those little pieces and further.
Right.
Then you have the very exquisite proteases that process hormones. They don't do anything but just take a prohormone, cut a very specific place in the protein, and then produce another one or two other little peptides that then have very specific activities. And so they run the gamut of being very, very fastidious about what they want to cut, to the ones like in your digestive tract, which are promiscuous. They just go out and chew up anything that's around.
So let's just bring ourselves up in the history on this. So 1984 then, we have via Gallo, the United States government claiming that it was a retrovirus that was causing immune systems to collapse by attacking CD4 T cells, the helper cells in cell-mediated immunity. So the researchers like you began looking for ways, how can we inhibit this replication of these retroviruses in the human body, assuming you believed, and which you did, which most of us believed at that time, that Gallo was telling the truth, or that he had found the truth. So there were two tracks. We were going along with the track of the AZT track approach and the protease inhibitor approach. Now, at what point did you become skeptical that we were looking at the wrong thing, that we were attacking the wrong villain?
Right. It was 1985 when I had my first doubts. There was a real problem there, and that had to do with the fact I was, again, in one of these Bay Area peptide meetings, and I listened. I was sitting next to two virologists at a Chinese restaurant, as I'd mentioned, and they were talking to each other. One of the virologists who was not working on HIV was asking questions of the other virologist who was, and he asked him. He says, "What is the viral titer?" And the viral titer means what is the amount? How much virus can you find in these AIDS patients? And then the virologist working with HIV said undetectable. And then I went, wow, that's amazing, because that went along with my notion of the Andromeda strain being very, very bizarre. There's something that we can't even detect, but yet it's killing people. And but then after about sleeping on it overnight, I realized, wait a minute, there's something wrong here. And then with more thought, I realized, well, they probably just got the wrong virus. When they were searching for the polio virus, they threw away 100 or two candidate viruses before they settled on what they call the polio virus. And I figured, well, okay, HIV is the wrong one. They'll have to just keep looking until they find the right virus. But then 1987 was the last year when I held any inkling or any belief or lingering belief that AIDS was as it was advertised, contagious, sexually transmitted, caused by a virus, so on and so forth. Because by that point, we've had 7 years of AIDS, and it was still only in these gay men, basically, in the San Francisco, L.A., New York. And it wasn't behaving anything at all like a contagious or an infectious disease. Women were almost totally out of the picture unless they were drug users and things. And so then, I started reading up everything I could on this AIDS stuff to see what was going on. 87 is when I knew there was a serious problem. And then I think it was 88 when I started reading books. I think someone down in L.A., Jed Adams and some other ones, AIDS Inc. and Scandal of the Century and all that sort of thing. And then I discovered this fellow named Peter Duesberg, this virologist. Remember, I'm a chemist. I wasn't a virologist.
I think he's mentioned in all the books.
I think so. And I have a huge library on this stuff, from those days. Now I've quit collecting everything because my library is big enough. And then I started reading. I read his 1987 paper, Cancer Research, where the world's leading retrovirologist, Peter Duesberg, published a critique of retroviruses in general, HIV in particular, about how these things could not be pathogenic, how they don't kill the cells that they infect. People worked with these things for decades as possible cancer agents, carcinogens, and that didn't pan out. And here it was, this World's Authority on Retroviruses, in this document, it's like a legal document, point for point for point for point for point, how HIV could not be causing this disease. And the fact that it was clear also confirmed my own view that AIDS was not a contagious thing, And then I thought, good, this guy settled it. He's got it all figured, so it's going to go away. All said and done, get on with other things. And, of course, here we are 25 years or so later, and that didn't happen. And it was towards the latter 80s that then I started talking to a lot of my friends and colleagues, UCSF and other places, and telling them about my concerns about this, the problems with the whole paradigm, the AIDS paradigm, contagious sexually transmitted HIV and all that. And it was a very interesting thing. People, because we had years of collaborating, working together, so we knew each other, and I wasn't a complete idiot. Probably they think I am now, but in those days they didn't. So basically it was silence. They wouldn't talk about it, and that was a very, very curious and peculiar thing for a scientist not to talk about something of anything. Scientists love to talk. And then over the course, I'm trying to shorten this story. After a while, I'd persist, and then I noticed that people wouldn't talk to me anymore. They wouldn't even just be around me, and I lost friends. And then I decided the only way I was going to keep some of my collaborations, the ones that I really wanted to keep, was that by mutual agreement, we would not talk about AIDS at all, period. Especially the guys I worked with on parasites, because I had a lot of good work going on with these guys at the time. So then I knew, then that's when my whole life went in a completely different direction. I realized there was something rotten in the Denmark of science, And then I started studying history, the history of science. I studied everything I can to try to understand what was going on with the psyche of my colleagues. Either I was absolutely nuts and crazy and I needed a treatment or something, or there was something really wrong with the whole edifice of science. And that's ultimately what I've come to determine and realize, and came across Peter. Now I'm a good friend and colleague of Peter, and many other people, over the decades, is that there's something really seriously wrong with institutionalized science. And I guess that's the answer to that for now.
Well, let's put this in some more historical perspective here. Now, that bringing us up to these late 80s, we were seeing gay men dying perhaps 2,000 or 3,000 a year, 81, 82, 83, 84, 85, 86, and then along comes AZT. Not the class of drug that you were working on, but this other class of drugs. And let's talk a little bit about what that drug started doing to people, some of whom had been diagnosed with presenting illnesses as having AIDS by the definition of opportunist illnesses, some of whom simply had taken gallows antibody test and were placed on AZT. What was AZT doing then given this paradigm that a retrovirus was causing it and Gallo had fingered the right retrovirus? What did AZT start doing to human beings?
Started killing them in droves. My friend, who's a physician in Germany, Klaus Kernlein, says AZT has killed a generation of gay men. And he's not the only one to say that. Not being a physician myself and having treated and seen masses of these gay men on these drugs, I've just seen them through meetings and things. You know, I can't state as forcefully as the physicians do. But it's AZT killed a generation of gay men in the late 80s, early 90s, and continues to kill people to this day. Not nearly as with the rapidity that it did back in the late 80s when they were giving people anywhere from 1 to 2.5 grams of AZT a day. Now they've lowered it way down and mixed it with other drugs, but all they've done is just lower the dose.
So let's now bring protease inhibitors into this timeline. When did we start seeing adding another drug, the protease inhibitors,
right,
to the drugs that gay men were being told they needed to take to get rid of a virus?
Right, and even before the protease inhibitors, because of the success in terms of commercial success and the FDA approval of AZT, that's when the other nucleoside analogs of that class, you know, DDI, 3TC, D4T, and the rest of them, were also in the works as nucleoside analogs. But those things are very toxic, as we all know. They all come with black box warning labels. They destroy organs. They all have their own spectra of toxicity. Destroy the bone marrow, the kidneys, the liver, the nervous system. They're DNA chain terminators, those nucleoside analogs. They are cytotoxic. They are designed to kill dividing cells, and even cells that aren't dividing. But they can destroy the mitochondria in cells. And just, extremely toxic things. Everybody knew this, and they still know it. And they were trying. The whole point of the protease inhibitors was to try to come up with a different class of inhibitor as a drug that would be much less toxic than the nucleoside analogs. And indeed, it is true. The HIV protease inhibitors are decidedly less toxic than the nucleoside analogs. In fact, if they were used to treat intentionally things like yeast infections, thrush, for example, the protease inhibitors are among the most potent antifungal agents I know. And if they were used at the proper dose, which would be much lower than they are used in HIV-positive people, they would probably be very safe and effective against fungal infections. In fact, back in the early 90s, 1993, I was at a Gordon conference where I saw for the first time the data on, I believe it was sequinafir, that was Roche's contribution of the HIV protease inhibitors, where that one and many others that I looked at, other competing ones, and these are some of the most potent inhibitors I'd ever seen in my life. They were beautifully potent inhibitors. They were what we call picomolar inhibitors, 1 trillionth of a mole. That's how, in a liter of solution, that's how effective those things were. Very, very tiny amounts of these things were effective against HIV protease itself. And so I remember in 93 thinking that if HIV, of course in those days I knew it didn't have anything to do with it, but I posed to the people, I said, if HIV causes AIDS, these protease inhibitors should prove that and they should cure it. That's how potent they were. But at that very same Gordon conference in 93, Roche had also revealed that they had completed a small clinical trial, which I believe it was 400 people, something like that, in each arm, those that did not get the protease inhibitor and those that did. And then they said they were ineffective. They didn't work. In other words, the people didn't live longer or they have fewer of the AIDS-defining diseases. And they immediately, at that meeting, then attributed the failure of the inhibitors to the virus mutating to inhibitor-resistant forms. Now, that's fascinating. At the very same meeting, they present the data of how potent these inhibitors are and the fact that they don't work clinically. And then that led me ultimately, well, I already knew the HIV was irrelevant, so then I started looking at how to convince my colleagues, who weren't stupid people, about the consequences of the failure of the HIV protease inhibitors. That led me to publish a paper, I believe it finally got published in '97, on how it was that their explanation didn't work. That if there were such things as inhibitor-resistant HIV, that the virus wouldn't be viable in the first place, and the drug would have already accomplished its goal, And there were actually a few scientists that, like I said, they weren't dummies, and they saw the evidence, they saw the kinetics argument and everything, and they realized it was true. That's why my paper even got published, because it went through the peer review process. This was before I was sort of blacklisted as being a denialist, as they say these days. But in those days, they went through peer review, and it flew, and they said, yeah, it worked. You're right, Dave.
That brings us to the next period of history now. So from about 1987 to the mid-1990s, we were attacking the alleged villain here, HIV, with these DNA chain terminators. We were moving toward finding other ways through protease inhibitors. That's when Dr. David Ho came up with this fantastical theory that there were millions and millions of virions being produced in the body every day, killing millions and millions of CD4 T cells. So we got to hit hard, hit early. We got to create a cocktail of drugs. Men had died, certainly, who were taking these high doses of AZT. Men had survived despite being on this chemotherapy. And suddenly comes this cocktail of drugs, the highly active antiretrovirals, another euphemism of the HIV AIDS industry, HAART. You got to have HAART. They were introduced, and I'm going to make an observation here that you can comment on, Dave. All these men, mostly gay men, who had been on this AZT or analogs of AZT were in really bad health, or they were dead. They were so far down that when these weaker drug cocktails came along, they were so far down it looked like up to them, and that was what has been called the Lazarus effect, that suddenly we've got this drug cocktails and we're no longer sick and dying.
Yeah, you left out one very crucial thing that happened during that period, and that was the introduction of the viral load test.
I was going to ask you about that.
Right. It plays a crucial role here. When you mentioned David Ho and all this other sorts of things. Remember, I told you in '93, and already reported that the drugs don't work clinically, the protease inhibitors. And people were still dying in droves on the nucleoside analogs. And they started reducing the dose of AZT in these other ones. They go from 2 grams to 2.5 to 2 grams, down to 1.5 grams, down to 1 gram, down to half a gram. Now I think with the cocktails, it might be 300 milligrams or a little less than a third of a gram or whatever.
Yeah, that's correct.
They keep going down on the dose, and as you keep going down on the dose, people don't get as sick. They don't make them as sick as quickly. You get chronic toxicities. You don't die so fast with these things. You can live long enough to get cancer because they're also carcinogenic. But so they stopped doing clinical trials, phase 3 clinical trials, which dealt with clinical outcomes like mortality, or does your disease, is it cured? Do you cure the specific AIDS-defining diseases? They quit doing that with the AZT trials, official studies. After that, they don't do those phase 3 clinical trials anymore, and they couldn't use CD4 cell counts as a surrogate marker, which they were using for a lot of nucleoside drugs, principally because of the Concord study, which really showed that the CD4 count is not a good surrogate marker for people's health. People can have high, low CD4 counts, be healthy, sick, or whatever in between. They just didn't work. They were all over the place. So since they're not doing phase 3 clinical trials anymore, where you actually look at endpoints like mortality, and the CD4 CD4 cell count surrogate marker is no longer legitimate, Roche, remember I said Roche was Sequinavir, that was a protease inhibitor. Well, Roche was the one that also acquired Kerry Mullis's from Cetus, his viral load, his PCR test. And Roche then used their brand new PCR viral load technology to come up with a new surrogate marker for clinical trials. And that's what David Ho latched on to, was to then tried to show some sort of correlation between administering his protease inhibitors as cocktails in these patients, and then watching a decline in their viral loads as determined by the PCR test. I think it was Dietls and some of these other guys that were using the viral load to show that people who were very sick had very high viral loads, people who weren't so sick had low viral loads, and they tried to correlate this with CD4 counts. And David Ho was the one who really used the viral load as the surrogate marker for the cocktails, for the HAART drugs. So then you give people these drugs, their CD4 cells go up and their viral load goes down. That's not an absolute, but in general that is true. And they were using these surrogate markers as indications and justification for using HAART. That they weren't following to see whether people live longer or better, but they were following their laboratory numbers. Are the CD4 cells going up when they give them these drugs, and does their viral load go down? And in general it does, but not very reliably.
And also, one thing, if you're doing a trial and the drug is reducing viral load, then is it not possible to terminate the trial early, maybe before you start seeing a lot of long-term side effects, claiming that you've reached success and you can't deny the drug to the people on placebo?
Oh, yeah, that's done. I cannot think of a clinical trial other than the Concord. The Concord went, I think, all the way to the end. And that was a European one, and there was tremendous pressure on those people to stop their trial before it was finished. But ever since then, as far as I can remember, every one of the clinical trials has stopped prematurely. It's like, I use the analogy, it's like watching a basketball game. The score goes up and down, and then at some point arbitrarily when your team is ahead, then you stop the game and declare victory.
That's a great idea.
That's exactly what they do with these, not only now with the HIV studies, but with cancer studies. They have one of the problems for healthcare in general in the United States, with the FDA and other places, is that AIDS now has set precedents that nobody in their right mind outside of AIDS would have ever considered. Doing away with phase 3 clinical trials, for example, using all these surrogate endpoints and all that. Now that's done across the board, and it's a very, very bad thing. And it's just routine. They consider it ethical. They use pull the ethical card on you and say, oh, well, these people are doing better. It's unethical to continue the trial, as soon as they start showing improvement. And unfortunately, this had a number of backlashes as a consequence of terminating these ARV clinical trials early. The public doesn't know much about this, but people in the business know. For example, like DDI, didioxyinosine, is extremely toxic drug. When it was approved by the FDA, the early studies were terminated very early on, and it looked pretty good. It didn't look very toxic. And then after it got approved, though, they were saying that just in a short time, a little bit longer than the clinical trial, people got extremely sick on these things. It was killing them really, really fast.
Sorry to interrupt, but in episode 13 of this podcast, we interviewed somebody who talked about the gay artist Keith Haring, who managed to get himself on DDI, and according to the person we talked to, Jim Forat, I think, he died within 2 weeks of starting the drug.
Yeah, that was a bad one. In fact, it had so much bad internal press. It was back in the days when I was still in the biotech industry, that people that always bring up DDI as really the way not to do a clinical trial, among my colleagues. I never saw it in print any place, but they would always talk about that. Premature termination of these trials, it's a serious, serious problem. It really drives me nuts, because the only way you can really argue these points is if other people are rational enough to agree with your point that you've made a good point. If people are going to buy into the emotional argument that it's unethical to keep doing this, when I say people, I'm talking about like the FDA or drug companies or anybody. There's nothing we can do. If they don't even accept the fact that premature termination of these trials goes against the complete notion of doing a clinical trial to begin with. The clinical trial is designed with certain statistical criteria so that you can answer the question with at least some reasonable certainty. Do these drugs that you give people, do they do more harm than good? Do the people that take them live longer or do better or stay the same or do worse than if you don't give the drug at all? And people always get better or do worse on drugs. They live or die with or without the drugs. That's why clinical trials include, hundreds of people or even thousands of people or tens of thousands of people, depending on the scope of the drug and the level of accuracy that you're trying, or confidence that you're trying to determine. It's because it's a statistical question that that you have to do it. And they know how to design these things right and everything. And now they're breaking all of their own rules. And if if they don't own up to the fact that they're breaking their own rules, then there's no amount of arguing. There's no hope for success to try to say, hey, folks, but you're breaking your own rules.
Now, let me at this point get us back on our historical timeline. So by the mid-1990s, we had found a new class of drugs to attack the villain, the wrong villain, and now we were essentially poisoning people more slowly. We were making people sick more slowly. Instead of all at once pounding their immune systems with AZT and killing them, we were now assaulting them with poison in the theory that this poison was killing this retrovirus. But we saw then people getting better. And you've already alluded to the fact that these protease inhibitors are antifungal, because some of the diseases that people with weakened immune systems may be fungal diseases. They're the DNA chain terminators are broad spectrum cell killers. Therefore, if you had some diseased cells, those cells are going to be killed too. So you've now had, for the last 15 to 20 years, a lot of gay men and a lot of black Africans increasingly being placed on these drug cocktails that in effect were a weaker poison than AZT, but which still includes analogs of AZT. Then bring us up to about 2006, when I think, correct me if I'm wrong, that there were studies that were done showing that the reduction of viral load, so-called viral load, was not in any way relative to increase in CD4 T cells. In other words, you could have an undetectable viral load, but you would have no greater increase in CD4 T cells, which was the whole theory behind why you were giving these drugs to begin with.
Yeah, I've actually collected quite a bit of literature on the failings of the viral load test. It goes back now all the way into the 90s, document in the mainstream zone literature of the failings of the viral load. And all you have to do is look at the insert that comes with the viral load test to realize that the FDA and other people were cognizant of the failings of the viral load test from the very beginning, because it says it's not to be used to confirm the presence of HIV, for example, and that's exactly what everybody uses it for. They say a viral load quantifies how much infectious HIV there is in somebody's circulation, in their blood, for example, and yet the inserts that come with the test say you can't do that. And many of the people who work with it know that that's true, that you can't use it that way. I think it was Deedles, for example. I put him on the spot. I think it was Deedles in one of these Gordon conferences, where he acknowledged that something like 60,000 to 100,000 of the viral load count, let's say, you may have only one infectious particle. In other words, only one out of 60,000 to 100,000 of these so-called viruses that you're measuring in a person may actually be viable or infectious. And then, so then I asked him, what about all this other stuff? And he said that the non-viable or the dead HIV is pathogenic. And in fact, people still are saying that. To this day, people are saying that, yeah, they agree that the viral load test is not measuring infectious virus in human being, but it still has clinical relevance. You know, it is still measuring a pathological effect of HIV of some sort.
The night of the living dead?
Yeah, it is. I know. It's all these zombies. But there was another thing that I think I might have sent you, David, recently, this Boosie paper, I think.
Yes.
I came across it. You're always coming across these things, where I've been following over the years the CD4 count that goes up, and it's been proven many, many different places, mainstream literature, that it's what's a so-called retrafficking phenomenon. The boosting of the level of CD4 cells in a person's blood after they start taking these HAARTs, these cocktails, is not. It has nothing to do with HIV. It has to do with the generalized toxic consequence of taking these drugs. Even chemotherapy for cancer will do the same thing. What it does, it causes a retrafficking of the CD4, even CD8s and other white blood cells out of your lymphatic tissue into your blood. And there's a boost. It's a non-specific general toxic consequence of taking these drugs.
And we measure, the AIDS doctors measure CD4 counts in the blood, in the serum. They don't generally measure them in the lymph nodes, right?
No, absolutely not. And then the other thing that I learned in the Boosie paper that was important to me was now they're documenting that the reduction in viral load, which is just a measuring of the RNA, messenger RNA that's in a person's blood, and they're trying to make it like it's HIV's RNA that's in the blood. It goes down somewhat in general when you take these drugs, but by no means absolutely. And they're saying, that means the drugs are working because the HIV viral load is going down. But it turns out it's another non-specific toxic consequence of taking these drugs. Messenger RNA in general from lots of different things, like your interleukins, for example, and other proteins that your body produces, the messenger RNA in your blood also goes down when you take HAART and these drugs. So now you have the elevation in CD4 counts as well as other white blood cells, and the reduction in viral load as well as the reduction in messenger RNA from many other proteins, going down as a consequence of these drugs, are both a toxic consequence of these drugs. It has nothing to do with HIV.
So, okay, let me get this straight. So the surrogate markers that we're measuring, CD4 counts and viral load, are a consequence of toxicity. So the better the numbers, the more the toxicity.
That's right.
And this paper was published in 1999. So
yeah,
it wasn't exactly yesterday.
Not that recent even.
Yes. So they've known about this for over a decade.
Oh, yeah.
And done nothing.
Right. And I was a little annoyed by those authors. You see this all the time in cancer research. I see it all the time with AIDS, where their own data is screaming at them. It's jumping off the page that there's a problem here, and there was no discussion. I couldn't find a discussion or concern that the depression of all of this messenger RNA in people taking HAART was a problem, of a non-toxic, of a generalized toxic consequence of taking these drugs. No, it's astounding how the people that work on AIDS, how they don't let the data speak to them. They're sort of like they have filters. They can't even see their own results. Like Padian, for example. Padian's a very good example.
Yeah, not only did she, try to do the best to deny her own results, she found no sexual transmissions, so she used rather less valid methods to estimate some amount of sexual transmission, even though she found none. And then later went on the internet and wrote an article denying totally what she'd found. And Rodriguez, they wrote the paper that showed there was no correlation between CD4 counts and viral load. Right. They did the same thing. They published their data. It was shocking. Then later they went on the internet and basically denied it all. And also another famous study is the Bruno study of IV drug users from the late 1990s, where in the same issue, they had articles trying to deny the results, which were that clean needle exchanges increased your risk of becoming HIV seropositive. It's a really bizarre phenomenon.
It's a general phenomenon. It really is.
So we end up with coming up to the present time, continuing to pump chemicals into the body of gay men and black Africans now to attempt to attack a villain, a harmless villain, a harmless passenger virus. And we see all of these adverse effects that are coming from it. But you talk to the person who's been doing this for 15 years and say, well, my viral load's down, and, I'd probably be dead if I didn't have yes, my face has wasted away, my stomach is distended, I have chronic diarrhea, but I'm alive. It's so hard to get through to people that they have been poisoning themselves.
Well, it depends. For example, I noticed in the gay community, because it is so entrenched, this AIDS thing is like a cult in a way, that they are more attached to the paradigm than heterosexuals, for example. Since the mid-'90s, because of the 1993 definition change, these two laboratory tests allowed you to be diagnosed with AIDS. There's more and more heterosexuals that get involved. And they're not attached so much to this paradigm. They're scared like everybody else is when they come back HIV positive or whatever, and they get pressured and frightened into taking these drugs. But because they don't have this mindset, this attachment to this paradigm, that they immediately recognize the toxicity of these drugs, and they acknowledge it and start talking to their physicians about it, and generally try to do something about it. So it's different there because they're not so brainwashed. They haven't been living with it on a daily basis. And there's more and more of this going on. So it's basically, it's not just on an individual basis, it's also more or less in a cultural basis, whether you live in Africa, Europe, the United States, you're gay or straight or whatever.
Well, as a gay man, I can tell you that when I attempt to have this discussion with gay men, middle-aged gay men, if you say, hey, this was not a retrovirus that was causing immune systems to collapse. It was our behaviors. It was our sexual transmission of old pathogens. It was our ingestion of toxins. It was the psychogenic effects of being a hated minority. That's what was causing immune systems to collapse. They will say, "Oh, Terry, you're dancing on the graves of our dead. You're in effect, you are you're not being a good gay man if you don't believe that it's a virus that was causing this problem as opposed to behaviors that was causing this problem." I have to tell you, I'm with Carrie Mullis, who said the sexual revolution and its results in immune systems collapsing was simply not a moral judgment. It was simply an experiment that went bad. But it really is so almost religiously, cultishly inculcated into the minds of the gay community that we simply don't want to hear it. And that is, of course, what we're up against. Let's move the conversation to something practical here.
Okay.
If you were a gay man who has been on these drug cocktails, who has been taking the protease inhibitors, who has been taking the DNA chain terminators, let's say 10 or 15 years, you're seeing the results, the facial wasting, the fat redistribution, and your body has, in some sense, accommodated itself to this slow poisoning. What would be your advice to this person? Go cold turkey, get off these drugs immediately, or do you have to sort of move slowly to get to avoid further toxicity by just immediately going off the drugs?
Boy, I tell you, you have to do that on a case-by-case basis. for me personally, from what I know, speaking, if it was me or anybody in my family or something like that, if they're taking these nucleoside analogs, it's like any poison, I'd say stop that immediately. You know, it's like cyanide or anything else. Why go off of a poison slowly? Then you have things like Positve, unfortunately, one of these anti-HIV drugs, that also has neurological, psychological, even psychotic consequences of it. People can get addicted to it, things like that. Then you have that problem, you know, if you go off of those things really, really quick, then you've got all these withdrawal symptoms and that sort of complication. So that's on a case-by-case basis. But I think my feeling is if you had to pick something, if a gun was held in my head, I'd say stop taking the drugs right now. That's that. The next thing is learn. Inform yourself about what's going on. There's tons of places. If you work at it, you can find out what's going on and inform yourself so that you can make right choices. So it's one thing that I would do in a practical sense, if you really, really want to get practical, I think we have to outlaw the HIV test. That test alone, if you get rid of that test, then there's no drive to drug people, because it's only HIV-positive people, certainly in the industrialized world, you know, by those tests, that then are prescribed the anti-HIV drugs. So if you stop the test and people don't become positive because they can't be tested anymore, then they're not going to be poisoned by these drugs. That's on a very practical basis. Unfortunately, we have the Centers for Disease Control now in the past year pressing for nationwide testing in the United States, for everybody in the United States to be tested for antibodies to HIV. And just yesterday, I saw in the Wall Street Journal about this, all the seniors in a class at the Urban Senior, it's called the Urban School of San Francisco. It was in yesterday's Wall Street Journal, where all 80 seniors, it's a small high school, have agreed to be tested for antibodies to HIV this coming Friday.
It's frightening. It is absolutely frightening the way that people are being pushed into testing. And I know this, sitting here in these 10 square miles surrounded by reality called Washington, D.C., that the political process is pushing, the funding process is pushing people into testing. The local HIV AIDS administration, every time it finds a new, quote, HIV positive, gets a couple of $1,000s more from the Ryan White AIDS pot of money. It's about $2 billion a year. Am I getting his name correct? The young hemophiliac?
Ryan White, yep.
Ryan White. Yes. The way the money is distributed is if a community is seen to have a high level of incidence of HIV positive, it gets more federal funds. The local HIV AIDS administration in Washington, D.C., the epicenter of this myth, has about 125 employees and a budget of about $165 million. I have to go back and check the records to make sure I've got my numbers correct, but it's something close to that. So in Washington, D.C., they push the testing in this majority black city because they get more federal funds pumped into the city treasury, and it subsidizes almost like overhead costs the wasteful city government of this of the jurisdiction we're in. In other words, basically, it's about money. It's about
the HIV test is the linchpin to this whole thing.
Yes,
it drives everything.
Psychological terrorism is what it drives in the gay community. You get the HIV test, you are psychologically terrorized, and then you're slowly, sometimes quickly, pushed into the drugs, and your life just falls apart.
Right. Just yesterday in the Guardian, UK newspaper, student and teacher unions are complaining about a plan to introduce HIV testing for 13-year-olds in schools. I mean, a lot of 13-year-olds wouldn't be sexually active. Can you imagine what would happen if you tested positive?
Yeah.
You'd be everybody would assume that you are promiscuous. Otherwise, how would you have got it? And you would be put on the quote life-saving treatments. Anyway, it's heartening to see that there's some real pushback about stigmatizing children at the age of 13 with both this label of promiscuity and death.
And then, of course, we're seeing all of this push to not only give people these drugs once they've tested, quote, positive, unquote. We're now seeing this pre-exposure prophylaxis, giving people drugs who have tested negative on the theory that it will protect them from becoming positive. The farther you go down this rabbit hole, the more bizarre it gets.
Yeah, that is one of the most disgusting and infuriating developments in a long time. It's this what do they call it?
Pre-exposure prophylaxis. PrEP.
Yeah, that's it. Pre-exposure prophylaxis. I mean, that is just only Hollywood could come up with something like that.
Well, it's kind of a nightmare. I look at these things from a point of view of money. If you just focus totally on finances, to make more money, you need to expand your market. If the number of people getting tested or becoming HIV positive in various ways is too low, well, you got to start giving the drugs to other people. I mean, and if they manage to give the drugs to every gay man and every African, well, they'd still want to expand their markets, and they wouldn't stop until everybody on the planet had protease inhibitors in their cornflakes at breakfast every day.
Well, every time you get a new HIV positive, you get a new candidate for the drug companies' profits. You get a new candidate for the city and state and county governments' revenue from the federal government, from the Ryan White Act, from the Housing and Urban Development Department, providing housing for victims of, quote, HIV AIDS, whether you're sick or not. If you get a positive test, it becomes a poverty program. It is absolutely stunning where we found ourselves now, I guess, 27 years since Margaret Heckler and Robert Gallo invented the AIDS virus at that press conference at a French restaurant in Bethesda, Maryland. I think we're coming up on our closing on our time here. Dave, just let me ask you a final question here. How can we move forward? What can we do to try to convince people that we had fingered the wrong villain and that the classes of drugs you were working on, that other was working on, are simply not helping people, they're hurting people?
Well, I think we are going in the right direction. We're doing probably the only thing that could be done is that we're not giving up. Rethinking AIDS now, especially in the leadership of David Crow, is far more vital and effective than it's ever been. There's a lot more people involved in it, with a lot more international interest. There's a lot of grassroots efforts, and especially a lot of this has been coming from the heterosexual people in the world because, like I say, they weren't brainwashed as much, and then when they start getting the toxic effect, seeing personally the toxic effects of these drugs, and how they're treated and the humiliation and intimidation, they start fighting back. Some of them, for example, like Carrie Stokely in Florida and other people around the world. The story is getting out there. The documentaries are being made, like House of Numbers, for example. That's a very good one, and numerous other ones. This program that we're on right now, and our 2009, RA 2009 is going to be an RA 2011. And there was this just last year in Vienna. These things are growing. I mean, it is absolutely growing. And there's another indication that we're going in the right direction, is that the attacks against us and against the critics of the contagious HIV hypothesis, these attacks against us are growing. That's always a good sign. It means that we are having an effect. There's more and more of these websites that are attacking us all over, and they're tracking us and monitoring us and everything. They wouldn't do that if we were irrelevant and if we weren't having an effect. So we are having an effect. Since you can't count on the government is the problem, clearly the problem here, so don't go to the government to fix it. It's got to be a grassroots solution, and the grassroots seems to be rising to the occasion.
Ideas matter and truth matters, and we thank you very much, Dave Rasnick, for your contribution to the truth on this subject.
Thank you. I've enjoyed it.
Thanks, Dave. It was a great discussion. It was wonderful. And thanks also to our listeners. Thanks for listening to another episode of How Positive Are You? And stay tuned for future episodes. And don't forget to check out our website as well. Goodbye.